So next up, I would like to invite Professor Dr. Castrén from Helsinki, who recently published the paper about the interaction of psychedelics and TrkB receptors. And this has caused quite a paradigm shift in the field. So I'm looking forward to your talk and learn more about the new insights in allosteric BDNF-TrkB signaling as target for not only psychedelic but also antidepressants. Thank you.
Thank you very much for the, can you hear me? Yes. Okay, it's better? Yes. So thank you very much for the invitation. And I would like to, based on the studies that I'm going to tell you, my colleagues and myself, we founded a startup company, but nothing of this, what I'm telling you, is about the products of the companies, naturally, because it's very new. So my lab has been working on neurotrophic factors, and particularly BDNF, for a really long time here. And BDNF is known to be a central regulator of neuronal plasticity. So it actually, when the neurons make connections and decide that this connection is very good and should be kept, then they release BDNF, and that acts on TrkB receptors, and then that initiates the sort of cellular process which then stabilizes that synapse. And BDNF here is binding to TrkB receptors, inducing its activation and phosphorylation, and then that leads the signal further in the nucleus. So this is what happens, and this BDNF is kind of a central regulator and actually sort of a mediator of the activity-dependent plasticity. So if you have two synapses here, if one is active, it will stabilize the synapse. The other one is active but much less, so then it loses in competition, and then it withdraws and goes away. So it's not just that you have more synapses, you need to have synapses that mediate activity in order for them to be saved. And the ones that are leaving, I mean, it's actually an advantage that they are leaving, because if they were left there, they would just increase kind of noise and not be useful for the system. So my lab found actually now, more than 20 years ago, we found that antidepressant drugs increase TrkB signaling by looking at phosphorylation of TrkB here. And we can see that drugs in many different categories are doing that. It took us then a long time to find out how that happens. But very recently, Plinio Casarotto, together with modeling people, we found that their TrkB receptor, when it in the transmembrane domains, that we usually always draw like this, they are parallel, but it seems that in reality the transmembrane domains are crossed like this. And when they are crossed, then there is a binding site for small molecule at this crossing, and we found that here we started with fluoxetine, labeled fluoxetine binds to TrkB in saturatable manner. And if we make a mutation in one of these amino acids which are critically predicted to be critical for this, there would be a much lower affinity for this drug. And then using a displacement assay, we can see that antidepressants in many different categories actually displaced fluoxetine from this site, indicating that they are binding on the same site. And what was actually a surprise to me and everybody, that ketamine actually also binds to the same site. And we have shown that not only by displacement, but also directly. And then hydroxynol ketamine, which is a ketamine metabolite that we have investigated quite a bit. Then Rafael Moliner is a very, very talented graduate student. He wanted to look at the effects of psychedelics, and we found that LSD and psilocybin also bind to TrkB. And if you look a little bit here, you can see that the binding site is a little bit outer, and the conformation of the TrkB receptors are a little bit different. But the big difference here is in affinity. Antidepressants in general, all the antidepressants, also ketamine, are poor ligands of TrkB. They have a low affinity, micromolar affinity. But LSD and psilocybin are very high affinity ligands. They bind here at about nanomolar affinity. So there's a big difference between these. And again, here, if we are making mutations, we have 2 mutations that are in this region where the binding is happening, and those greatly reduce the affinity of these compounds to TrkB, whereas a mutation which is in the transmembrane domain but is outside of the binding domain, that has no effect on the binding. So the model that we have come up here is that the position of these TrkB transmembrane domains is dependent on membrane thickness. So, and membrane thickness is largely determined by the concentration of cholesterol in the membrane. So usually you think cholesterol as something bad that you get when you eat burgers, but actually it's a major constituent of the plasma membrane, and synaptic membranes, the concentration of cholesterol, molar concentration of cholesterol is up to 50 or 50%. So every second lipid in the synaptic membranes is cholesterol. So when there is more cholesterol and TrkB and the membrane getting thicker, then at some point this dimer type of structure falls apart, and then as a consequence, TrkB is not retained in these synaptic membranes. But if LSD or other ligands here, if they bind to this conformation, they act as kind of wedge to prevent this flipping of the receptors into the single molecules and getting out of the plasma membrane. So they retain the presence of TrkB in plasma membrane, and we have shown that actually happens. And when it is retained at the plasma membrane, the probability that its ligand BDNF comes and binds to the receptor is increased. And we think that that's the mechanism how this is happening. So these molecules are not agonists of TrkB. They don't on themselves activate TrkB. They increase the sensitivity and probability of BDNF to act on its receptor. So here you can see that we have looked at very low concentrations of BDNF here, and then we have this TrkB Fc, is a molecule that traps BDNF out of, this is in vitro, from the medium, that prevents it from binding to its receptor. So if you add BDNF and at the same time with this trapper, then there is no effect. If you add LSD together with this trapper, also the effect is gone, because it is dependent on BDNF, and combination of these is then also not acting. And here we have then a spine growth assay, something that Gil Dolan was calling artifact earlier today. In my mind, there is certainly a place for in vitro studies here, because they look at the mechanism, and the same mechanisms are then later in adult brain used for example, for the opening of critical period plasticity. So this is not representing critical period or critical period plasticity. Anyhow, what we find here is that again, LSD is increasing the number of spines, and also psilocybin. This is psilocin. And then this effect is not, in our hands, blocked by the antagonist of 5-HT2A receptor, but it is blocked by this BDNF trapper, the TrkB Fc. We have then made a mouse that has this one mutation in this critical tyrosine that prevents the binding of these drugs. The mice are otherwise completely normal. We have done several behavioral studies there. They also bind BDNF. BDNF is inducing the activation of TrkB. The only difference is that drugs don't bind. And what we can see here is that if you look at the immobility, then LSD is producing an antidepressant-like response in reducing immobility, and then this is not blocked by the pre-treatment with NMDA receptor, 5-HT2A receptor blocker, but it is blocked in the mice that have this mutation. Then we have looked at head twitches, which is kind of an indicator of the experience, psychedelic experience, considered to be this. So what we can see that in wild-type animals, if you give LSD, there is a robust response. LSD response. And this response is completely normal in the mice that do not have this, that do not bind LSD to TrkB. Whereas if you then use an antagonist of 5-HT2A receptor, you can completely block this effect. So these kind of data suggest that there are two effects, one which is 5-HT2A mediated and would be correlating with the experience, psychedelic experience, and then there is another which is which is TrkB mediated and would be correlated with plasticity and antidepressant-like effects. Gül did a great job at describing critical period plasticity. My lab has also been using critical period plasticity, especially in the visual cortex, for a long time. And I don't have to go there because she was explaining this for some people that were not here. And the kind of paradigms that we are using is that during the critical period, if you close one eye in a mouse, then or human for that matter, then the competition that is ongoing because of the enhanced plasticity during critical period leads into a situation where the open eye is taking over the input into the visual cortex, the closed eye loses it, and then there is a shift in ocular dominance that happens. So normally you would be here in values, and if you don't close the eye, that would happen. And at the peak of critical period, if you close an eye, there is a shift in critical period. That doesn't happen in adult animals because the critical period is closed. So if you had one eye closed for 1 month, and then for some reason, then you opened it, you would blink a little bit and you would still see. But a baby, if that happened in 1 year old, that would have a tremendous impact on the visual cortex. So we use that as a model. So we looked at adult animals, and we thought that if you increase plasticity by antidepressants, you might actually see some sort of switch in ocular dominance. And that actually happened, but it was not a small switch as we expected, but it was a large switch, indicating, and also other characteristics of critical period of electrophysiological and histological characteristics indicate that there is a full opening of critical period like plasticity in the visual cortex. If you don't close the one eye, then nothing happens. So the drug itself is producing no change in the visual cortex. It only opens the plasticity. The effect is only becoming apparent when you close one eye and change the environmental input. This is actually really important and also was emphasized by Gül earlier, that you need to have this change in activity pattern in order to make the plasticity, which is increased plasticity in the networks, visible and functionally relevant. We also did a study with looking at whether you can recover the function of networks that are initially made wrong. So we had a mice, these were rats actually, and that was done in a collaboration with Lamberto Maffei's lab. The eye was closed from the early on until adulthood, and when you do that, then one eye was weak in vision. And if you open that eye, and even if you patch the better eye, forcing the mice and rats to use the weaker eye, vision does not recover. But if you treat them with fluoxetine at the same time as you do antidepressant or fluoxetine at the same time, you fully recover. It is important to emphasize, and I can actually show here, that for the treatment that we have, fluoxetine has to be given for a long time. If you just give it for 1 or 2 days, it doesn't do anything. You need to give it for, here we have given it for 21 days, and then we start the effect. And here is a switch. But then with ketamine and its metabolite, hydroxynorketamine, if you just give it just before, and we have given it actually here 3 times, then you would actually get a full switch in ocular dominance. And also LSD, we just give it 1 time, and then you would actually get an effect. So all these compounds increase and open a critical period plasticity. So somebody was asking a question whether this now means, what does that mean if you look at the visual cortical plasticity to many other effects? So Gül was showing that similar kind of things with these drugs is happening also in social network. From my mind, if you take a drug that goes everywhere in the brain, so all the systems that have these characteristics and molecularly are able to respond would increase their plasticity. So what is the behavioral outcome of depends on how do you change the input activity. If you change it in the visual cortex, then you would have a response in visual cortex. If you do that in social domain, then you have a change in social domain. So I think that there is, and the model of visual cortex as a model is very good for two reasons. First, because it has been investigated intensely by many people, and Nobel Prizes were given for it already in the 80s. And the second reason is that you can easily manipulate the environment by just closing the eye or put lights off, or for that matter. So we have then used also mice that have this mutation in TrkB receptor and fluoxetine, and we have done that also in ketamine. The effects are lost. So again, I would like to emphasize that BDNF and behavioral, they are normal, and BDNF has its effect. It's just that drugs don't bind. That indicates that this effect is mediated by these drugs binding to this particular site in TrkB receptor. We also then looked at the effects of, we're interested in what is the cell type in these networks that would mediate these effects. And we were particularly interested in parvalbumin-containing interneurons, because it has been shown that they are immature during the critical period, and they don't effectively inhibit the pyramidal network. But then when the critical period gets close, they mature and they become more effective. And upon the critical period, you would actually have an enhanced in inhibition. Also, these parvalbumin interneurons are encased into what is called perineural nets. That's an extracellular matrix components that are known to also enhance the inhibition, and then thereby reduce the reactivity of the visual cortex, pyramidal neurons. So what we did is that we made a mouse where TrkB receptor was removed out of this parvalbumin interneuron specifically. And if you do that completely, the mice have a balanced phenotype. You cannot investigate there. But if you use heterozygous mice that have 50% of the TrkB receptor there, that's what we have used here. And what happens is that if you close one eye in an adult animal, there is a shift in ocular predominance when you have fluoxetine there. But in these mice that have reduced TrkB in parvalbumin interneurons, you don't see that. Reversely, we then also made an construct where TrkB receptors can be activated by light, specifically by inducing their dimerization through this light-sensitive PHR domain. And when we express these specifically in parvalbumin interneurons, these constructs, so then only the parvalbumin interneurons in the visual cortex can be activated by light. And this is done by Friederike Winkel, when she was shining light 30 seconds twice daily, morning and evening, through the skull into the visual cortex. And that would be, and while the eye was closed, then that would induce the critical period like plasticity and shift in ocular dominance. And that indicates that these receptors here, TrkB receptors in the parvalbumin interneurons, are critical for this induction of critical period plasticity. We also have evidence that these perineural nets act by inhibiting TrkB activity within the parvalbumin interneurons. And if you disrupt this perineural net, and you actually can induce that by critical period like plasticity, then the TrkB receptors are activated, and that leads into similar kind of situations that you have here. So, sort of as a summary, there is now a consensus that the long-term effects of psychedelic compounds and antidepressants are mediated by changes in the plasticity, and there's also consensus that TrkB signaling, BDNF-TrkB signaling, is critical for this plasticity. Where there is not so much consensus is where the TrkB signaling actually is in this cascade. It has been considered that it is kind of downstream effect, that is induced by 5-HT2A receptor or NMDA receptor receptors. But now our data suggests that it actually is a direct effect. So this combines fluoxetine, ketamine, and psychedelics. They directly bind to TrkB and allosterically modulate. So now allosteric modulation means that they are not themselves agonist, but they help BDNF to more effectively bind it. So what we suggest is happening is that by binding to this receptor, that you have an increase in TrkB in synaptic sites, but that receptor is only activated in those synapses that are active and where BDNF is being released. So those synapses that are not active, even though TrkB receptors are there, they will not have increased signaling, TrkB signaling, and that would lead to retraction. So, and as I said in the beginning, this is important and critical for the activity-dependent synaptic plasticity, and that these drugs actually enhance and sharpen this activity-dependent plasticity, selection of the active synapses at the expense of inactive synapses. And then, as a consequence of all this, you would actually get an enhanced plasticity that can then, if you provide guiding behavior, then can change the structure of the network towards normality or towards beneficial effects. But that does not have when you don't provide any activity. So this is something that we have been doing in mice. And most of that, some of them in rats, but not really in humans. We have looked at human receptors, and they work the same way. But I think this is a big sort of disclosure here. Additionally, I think that figure also indicates and reminds something that Gül Dölen was also emphasizing previously. You need to provide the, at the time of enhanced plasticity, you need to provide the positive experiences. And one of the reasons why psychedelics and ketamine seem to be so much more effective than regular antidepressants might be related to this, that they are administered in the presence of a professional, especially psychedelics. This is intentionally done very supportive. In the case of ketamine, it is not so supportive, but nevertheless, patients are supposed to go to the clinic to get these drugs. Whereas SSRIs, you get a prescription, you go home, regardless of what happens in home. It might be a mess, or it might be a supportive environment. And then what is the outcome? It will depend partially on that. So here are my conclusions, and what we have seen is that psychedelics bind to TrkB receptor with high affinity, and then other antidepressants with lower affinity. And they are allosterically activate BDNF signaling, so they are not direct agonists, but they increase BDNF effects. And that leads into the reactivation of critical period plasticity that we call I-plasticity, and this is the same thing in my mind that Gül Dölen is calling metaplasticity. And an important thing is that this I-plasticity need to be combined with active therapy or some sort of training that brings about the physiological response. And the data that we have produced and that others have produced is kind of a message for this community that there is now a real possibility that the effects, the experience, psychedelic experience, and the long-term plasticity and clinical effects are two separatable things. And if they are two separatable things, then I think that everybody needs to be thinking, what is it that I'm interested in? What is it that I'm studying? Am I studying the experience, or am I studying the long-term effects? I don't say that the experience would have no role in the long-term effects. I think there certainly we need much more research, but it is a possibility, and I think that this has to be sort of teased apart in human studies, because in mice, there already is some data. I think that where awaiting human studies testing these hypotheses. Thank you very much.
Thank you.
Sorry, I got carried away. I forgot to acknowledge the people that were doing this. So Eero Castrén here, he was involved in looking at the antidepressant effects. Right now he's in Elsevier rejecting the papers that you are trying to send him. And this is Raphael Moliner, who was largely involved in the psychedelic work. And here are our funding bodies.
Okay, so I start with the questions. I will go around past the microphone.
Thank you for the fascinating presentation and the groundbreaking discovery. Really, to me, it brings how there are these two waves for the psychedelic effect, therapeutic effect. The experience with the insights and then the neuroplasticity to really learn, integrate. And now there are psychedelic experiences with similar phenomenology without this external substance. And I wonder, what's your view? What would be the endogenous ligand?
So I think that's a fascinating question, because I think we know very little about the neurobiology of these non-pharmacologically induced experiences. And I have no response to that. I mean, there is no, as I said, I mean, that kind of things are, if mice have that at all, and I doubt that they have, it may be human specific, because also mice produce just this head twitch response, which is not very sort of face valid for humans. So it may be actually difficult to investigate. But I think the question itself is very important and interesting.
Once more, thank you for your presentation. I'm very happy that you bring Casaroto's work into the field of psychedelics. I saw his paper a couple of years ago, and I got interested in his work. And I also saw that this mechanism is not available until the second week after birth of rats, which for me is very, and somehow maybe it's kind of linked to your question. Which is outstanding for me, because this is right the moment where a nation paper back in the 70s detected that endogenous DMT levels in rats goes up, rise, and they reach like a top, and then they go down to mature levels. So I know that this topic of endogenous DMT is controversial, but wouldn't it be reasonable that this mechanism that is available just in this very precise moment could be like somehow mediate until some extent this critical period naturally occurring in rat pups, and that maybe endogenous DMT is playing a role in all this?
Yeah, first, thank you very much for reading our work very carefully, because he's referring to our old paper where we were looking at and this is done in vivo. So we were looking at the phosphorylation of TrkB receptors in response to antidepressants at different ages. And to our surprise, we could not find the phosphorylation of TrkB early on until 2 or 3 weeks. And then there is an increase that, with the drug. A very curious thing about this is that we actually in this study also replicated all the findings that everybody actively wants to forget, because nobody can explain it. And this is that phosphorylation of TrkB in response to BDNF is very strong in early life, in embryos and early life. But at about the same time, at about 3 weeks, it completely disappears. So if you add BDNF onto slices of adult animal, there is no TrkB phosphorylation, and nobody knows why that is the case. So, but there is a switch which may not be causal, but it is correlational, that first you have TrkB as a BDNF ligand, and then antidepressants. Now the question about endogenous DMT, I cannot respond. I have no idea about that.
Yeah, thank you very much. Fascinating talk. I was just wondering about the timeline. Maybe you can tell me about that, of this TrkB effect on plasticity, because you said it can only work in the presence of activity provided, like positive activity, for example, or the presence of a therapist. I was wondering if this is just this combination that makes it unique, like the psychedelic subjective experience. Would it be possible that this is the very activity that is provided on a subjective level that makes the difference between, let's say, the strong effect and fast effect of psychedelics versus the antidepressants, because it is just so unique as experience, like ego loss and all that, and getting rid of the ruminations, that this, in combination with ongoing TrkB plasticity, just provides what you need for this stunning effect. And it could be found out if you, yeah, I don't know if there's any trial that has tested that, give psychedelics at the same time of, let's say, ketanserin, to, blocking the subjective experience for a therapeutic use.
Yeah, so this is, you know, these antidepressant psychedelics, if you look at, you know, I used to be, early in my life, I used to be a pharmacologist, then I quit. But, you know, at some point I was teaching pharmacokinetics and models of what happens. And they say that drugs can only act when they are present. And now we have these, you know, with antidepressants, we have drugs like classical antidepressants. For the first 2 weeks, they are present, but they don't have any effect. And then you have ketamine and you have LSD and psychedelics that they are there first high and then they go away. But then like several days or up to weeks and even longer, they still continue to have an effect, even though they are not present. So that part is, it's not related to TrkB phosphorylation, because we know that the phosphorylation is also transient and goes away when the drugs go away. What actually, so then, and this plasticity is considered to be the kind of key thing that then produces these long-term effects. I think that we still have, we're missing something, you know, explanatory between there. I mean, the drug effect and how does that really then, for the long-term effects, happen. You're then referring to whether the experience could actually be important. And I tried to say that in the beginning. I think that this is completely possible. We just don't know at the moment. And I think that we can either use inhibitors, but most of the inhibitors are not that great at the moment. They are quite unspecific. Well, I mean, at least Olson is developing now drugs that would be not having the sort of psychedelic experience, but would produce plasticity. So I could actually, I mean, I think we can envisage three types of drugs. We would have a regular antidepressants that work for certain amount of people. We could have new drugs that rapidly induce the activity of TrkB and plasticity, but do not produce a psychedelic experience. And then people who are not responsive to this, then we could have a third, which is the more classical psychedelic that would produce this behavioral effect that could potentially very well enhance the plasticity and direct and guide the plasticity that is produced at the same time. So I think that these are the kind of questions that we need to look at in the future.
Yeah, thank you for the cool data. I think it solidifies quite clearly that TrkB is very important in the effect of psychedelics. As you might be aware, there's also a bit of a discussion about the druggability of TrkB. There was some discussions on Twitter and in some Slack groups, some people showing that depending on the methodology you use, a lot of the things we consider to be TrkB inhibitors or activators actually are not. And it basically, I'm not saying that it questions the data, but it makes me a bit more critical, and I wondered what you can comment on this.
Yeah, so from the point of view of drug development, this has been a nightmare. I mean, TrkB and TrK receptors, because all the big drug companies, have spent a lot of money on trying to look at TrkB agonists and using especially signaling pathway assays. And they have been pretty much negative. So there are some compounds that are now there, and their specificity and is also questioned. We have also then done similar kind of screens, and they have not worked in our hands. So now the binding site that we have now is quite different, also because it is allosteric. So you would need to have the BDNF effect there as well. You would not, if you just screen in the cell line, they would not expect it to produce any positive results. Altogether, this is, in many ways, these are early days. So, we are very confident with our results. We have done them with many things. We need to have other labs to replicate them, and they need to replicate them in the way that we did them, not the way that drug companies have done them and failed. So I think that you're completely right that science is the kind of thing that you bring results, and the data is something that I have always sort of learned to appreciate, not the speculations. And then over the time, you need to have replications, and you need to have more mechanistic data that would then sort of consolidate or then invalidate this hypothesis. So that's what science is about. Thanks.